Thank you so much for attending today. We're so excited to learn more about this pest that seems to have just popped up in the southern United States. The title of the webinar is Understanding Pasture Mealybug, a new invasive pest of pasture grass, turf grass, and sugarcane in the South. Of course, it's new for the US, right? But it's been other places for quite a while. So we're really excited to have some speakers that have been dealing with this for a while and that can share their insight, especially because we don't have very much. I am Kayla Watson. I'm the Communication Director of the Southern IPM Center. And at the Southern IPM Center, we present research issues and programs in integrated pest management from the Southern region through webinars. And we're so excited to be able to host this today. We are housed at both NC State and UGA. We have a mission to coordinate IPM across the region. And the regional IPM centers are supported by the USDA NIFA Crop Protection and Pest Management Regional Coordination Program. Any opinions, findings, conclusions, or recommendations expressed in this webinar are those of the authors and should not be construed to represent any official USDA or US government determination or policy. For the webinar today, we are so excited to be able to organize this talk with several different places. So USDA ARS, the Queensland University of Technology, the Florida Department of Ag and Consumer Sciences Division of Plant Industry, and of course hosted by the Southern IPM Center. So the first speaker for today, the first speaker of two, is Dr. Erin Powell. And Erin is a biological administrator and the scale insect taxonomist for the Florida Department of Agriculture and Consumer Sciences Division of Plant Industry. She serves as the curator of scale insects for the Florida State Collection of Arthropods, estimated to be the third largest scale insect collection in the Western Hemisphere. So that's pretty awesome. The research broadly focuses on mealybug and scale insect taxonomy, invasion biology, and predator-prey interactions. And since Pasture Mealybug arrived in Florida, she has been working on its taxonomy, life history, and natural enemies. So, Erin, thank you so much for joining us today. And we look forward to hearing about the Western Hemisphere part of the Pasture Mealybug.
Hi, everyone. Thank you for the introduction, Kayla. A lot of you have probably heard me speak, and I really want to get to Carrie's part quickly. So I'm just kind of zoom through what we know about the pasture mealybug invasion in the Western Hemisphere that has happened within the last couple of years. All right, let's see. Slides to progress. There we go. Okay. So pasture mealybug is in the order of the true bugs, the hemipterans, the piercing, sucking mouth parts. They tap into the phloem of their host plants. And the one in question that we've been talking about is Heliococcus somervilli, which is also known as a pasture mealybug, which is responsible for pasture dieback. This is a grass specialist mealybug, so it only is feeding on grass hosts. The damage starts out usually as a yellow and then rapidly progresses to being red or purple. But in a lot of grasses, like the sugarcanes and guinea grasses, we're finding that they just kind of go yellow before the leaves senesce. They don't necessarily all have the same damage pattern. So just some examples of within Florida, some of the damage that we've seen early on. Limpo grass was the first things that we saw that were affected. You can see the red out in this field of limpo grass here, and then some of the biomass that's died back with the dead leaves here. And these other closer up photos, you can see that there are heavy infestation with the white bugs there on the undersides of those leaf blades. So in contrast, in sugarcane, we see that the leaves are going yellow, chlorotic before senescing. So a little bit of history of the pasture mealybug. Somerville in 1928 was describing this attack on grass in Eastern Australia. And at the time, the mealybug did not have a scientific name. So this kind of original paper talks about the damage and some of the control measures that they used back in this first great incursion of pasture dieback in Australia. Fast forward to 1978, Helen Brooks found the specimens from Somerville in the collection and also realized that they matched some specimens from Pakistan and described the species as Heliococcus somervilli. In 1978, Ghosh and Ghosh described what would become a synonym. So they described another species from India that ended up looking about the same. So Douglas Williams from the UK synonymized them in the early 2000s. So he basically said, I think that they're actually the same species. Then 1998, there was a pasture dieback outbreak in New Caledonia. And then around 2012, Carrie can correct me if this is incorrect, but around 2012, it seems like they started having these kind of symptoms of pasture dieback in Eastern Australia again, but it wasn't actually attributed to the mealybug at first. It took a little while to figure that out. So then fast forward to around 2020, 2019, there started being some pasture mealybug popping up in the Caribbean. So the USDA APHIS intercepted it a couple times in quarantine from Puerto Rico and from the British Virgin Islands. And then in Barbados, it was reported to be attacking lawn grasses, pasture grasses, and that was reported in 2020. So you've noticed that I've kind of marked things in red and blue, and this is corresponding to the different variants, which essentially the new variant does not necessarily match the morphology of the species as described by Helen Brooks from the early invasion described by Somerville and then the India and Pakistan. So what I have is coded blue for old variant and then coded red for new variant. So in 2025, Texas first reported pasture mealybug. They estimate that it probably was there around 2022, and this has kind of been confirmed by some grower crop consultant photos that may indicate that it was actually there earlier. Early damage was thought to be the Rhodes grass mealybug, Antonina graminis, which is another grass specialist mealybug that actually looks quite different. If you get up close, they're dark-bodied and really purple with no legs and very stubby antennae compared to pasture mealybug, which is a much leggier mealybug. So now we had a new variant, also was reported from Mexico around that same time, that kind of like summer, July-ish 2025. And personal correspondence with the folks in Mexico indicate that that is also new variant with the morphology. In early 2026, Louisiana first reported it attacking sugarcane there. And in Florida, we got our first detection on May 27th of 2026. And we also have the first record from the state of Georgia as of just a few weeks ago. So now looking at 4 states within the southeastern United States. Our current Florida distribution is now at 24 positive counties. We have been extensively surveying throughout Florida to try to get an idea of how widespread pasture mealybug is. To kind of give a context, from May 27th, we were on the ground surveying the next week and had confirmed 9 counties positive within 4 days. So it was clearly there and had been spreading around for a little while before it was first recognized. And in Florida, that included not only pasture grass and grasses that were being purpose grown for cattle forage and hay, but also all of our sugarcane growing counties around Lake Okeechobee were already experiencing a dieback due to pasture mealybug at that point. So in good news, we do have early evidence of parasitism in Florida. So this is a second instar of pasture mealybug that has a larva of a hymenopteran slide mounted inside of it. We are also seeing evidence of parasitism with these dark spots on live bugs where wasps have oviposited within them. And then we've reared out a few initial natural enemies, inserted wasps from the different variants. So again, I really want to give the floor to Carrie and let us all kind of glean knowledge from her many years of working on this pest. But I did just want to let you know that I'm going to be talking more about the taxonomy of pasture mealybug and the 2 different variants and the differences in the morphology and the molecular this Friday with the Everglades Research and Education Center, and that's also going to be available on Zoom. So just a quick plug for that if anyone hasn't had enough pasture mealybug already. So thank you, everyone, and I will let Kayla introduce Carrie now.
Thank you so much, Erin. So, Dr. Caroline Hauxwell is an associate professor of insect ecology, pathology, and integrated pest management at Queensland University of Technology in Australia. She and her team have studied the Pasture Mealybug in an ongoing outbreak in Australia over the last decade, so 10 years. And she's going to share insights into its biology and potential management. She is a graduate of Oxford University and Imperial College London with over 40 years experience in tropical and subtropical pest management and biosecurity. So thanks again, Carrie, for coming, or I'll call you Caroline here, but thank you so
Yeah,
much.
whichever is good, yeah. Thank you. Well, thank you all. And I'm going to move straight into slide sharing because I want to get on with this. And we none of us want to be here all day. So you've already shared my slides. No, you haven't. Okay, hang on. Share screen. And we should be good to go. Tell me when you're up.
Looks good.
Good. Okay. And if anybody's into literature around this, there's a science fiction book from the 1950s called The Death of Grass, which is what I'm referring to here. It's quite dystopian. We've been battling with the mealybug in Australia. It probably showed up, as Erin said, in around 2012. And it really started to move quite extensively from 2015 onwards. Um, as Erin said, it was first detected in Curroy in Queensland in about 1926 by Somerville, and has been reported in several other places since then, including Puerto Rico and Barbados, and now in the USA, and found on sugarcane and rice in Pakistan and West Bengal. The recent outbreak is a new variant. The old variant was found by Somerville and in Atherton in 1938. In 2016, Desley Tree and Mark Desley Tree described a mixed population of old and new variant from Atherton on a single blade of grass. And there have been outbreaks subsequent to that, in New Caledonia and Puerto Rico. And we found mixed populations of old and new variant in Kin in Queensland, very close to Curroy, where Somerville found his original isolates. The new variant is similar to the Pakistan variant that was found on sugar, but it does have some subtle differences. It was described in this mixed population by Desley Tree and Mark Schutze in 2016 and is the cause of the ongoing outbreak in the USA and Mexico. We've just got a paper, it's now in press. It's been accepted by Journal of Economic Entomology. Ina Hernandez Europa led this paper. Basically, it combines the morphological comparison and the molecular comparison. And there are a few differences between this variant and the holotype and paratypes. The majority of samples that have been collected from 2015 onwards differ from the 1926 paratype. They particularly differ in the presence of pores or absence of pores in conspicuous nodules on the hind tibiae. And the type new variant has these pores absent. There are other smaller differences, which I list there between the type A, which is the old paratype, holotype version, and the new variant. But the main difference and the most easily recognized is this absence of hind tibia pores on the type B new variant. We've done some systematics on this. Jen Dixon and Ina Hernandez Europa did the majority of work on this, sequencing 18S and 28S from both the old and the new variants. We were very lucky, and I mean lucky, to find two specimens of the old type in our collections. We spotted them first in the molecular data, and then we were doing a non-destructive sampling method, so we were able to send the cuticles to Mark Schutze at Biosecurity Queensland, who identified them as consistent with the paratypes. And you can see from the 18S and 28S sequences that those two samples that we found do not consistently differentiate with or align with the old variant when we look at the 18S and 28S sequences. we were only able to amplify dynamine from one of the old specimens, and it was clearly different from the B type. And if you're looking in your American populations, you have more of the old as well as the new. Dynamine would be one to look at. But I'm actually a microbiologist who works on insects. And I was very interested in the bacterial symbionts in these populations, especially as it might relate to virulence. And Ina sequenced the 16S V3, V4 regions for the symbionts from the 2 specimens and from 25 specimens of the new variant, and found that they all have 3 distinct subpopulations of the main bacterial symbiont in them. It's an obligate symbiont called Tremblaya fenicola. We only got 1 read of the main dominant variant from the old type because one of the new types we had very little DNA left. But within the new type, they all have the same 3 variants, 1 dominant variant and 2 subvariants. And within the old ones, they both have the same dominant one, and one of them has 2, again, totally different subvariants of the symbiont. So on the basis of both the morphology and some of this molecular biology, we're saying that they're new variants. We aren't at the point yet of saying that they are different species. They certainly have differences in biology, and the new one is far more virulent than the old one and far more damaging. And there's a paper just been submitted by Z. Ahmed from Clemson University looking at cytochrome oxidase 1, and that's coming to a very similar conclusion that this needs a full taxonomic revision before we're going to be able to say whether it's a new species or not. But it is certainly a new variant. It can be differentiated from the old one quite readily, and it is extremely damaging and very fast spreading. The range in Australia now, I hope you can see the arrows on the screen here. It was first found around Brisbane, but the new variant now is out at Longreach and Mount Isa, and as far north as Tolga in the Atherton Tablelands, and down to the Hunter Valley just north of Sydney. You can see from this map of the Köppen-Geiger climate classification that it's occurring in these warm and hot grassland areas, temperate areas on the coast. They're really subtropical, up into the subtropical areas around Tolga, and out into the arid drylands, which are very important pasture country for Australia, but also creeping south now into the more Mediterranean climate areas as we get towards Sydney. And that's quite a wide range of climate. It's also still moving. It's still moving west from Mount Isa, and it is still going to be moving south from the Hunter Valley. So we don't yet know the full climatic range of this problem. If you look at that in a world sense, these are the India and Pakistan populations up here, again in those sort of arid grassland and subtropical and tropical regions. The Queensland areas, New Caledonia over here on the far right-hand side, which is a sort of tropical island climate, and the Caribbean, of course, which is leaving quite a large area of the southern USA and Mexico quite clearly with climatic conditions that are suitable for this mealybug, as far as we know, but also large parts of Europe and some of North Africa, which are also climatically highly suitable, we think, to this pest, and indeed in Southern Africa and South America. And I'm extremely concerned about how rapidly and damaging this pest is when it can spread into these regions. It has a very wide house range, some of which are extremely susceptible: creeping bluegrass, which is Bothria chloa, and Chancrus ciliaris, which I believe you have as well, but also broadleaf passipalum and sugarcane. And we have done tests with sugarcane that have shown that it is a highly susceptible host. The mealybug can be managed quite well by use of imidacloprid, And we think that one of the reasons we don't see as much of this mealybug in Queensland sugar, though it does occur in Queensland sugar, is that there's a lot of imidacloprid used in Queensland sugar production, mostly for greyback cane grub, but for various reasons. There's a lot of not just introduction in the soil at planting, but also foliar sprays. And we think that's actually been keeping it under control in sugarcane in Queensland. There are also several grasses which are less susceptible, and notably it's also taking out a sedge here. We're not the first to report it in sedge. It was also found in sedges in New Caledonia. And several native grasses as well. This curly Mitchell grass is a very important dry grassland native species, and Themeda down here, kangaroo grass. But there are also several varieties of grasses that are much less susceptible, including Mekong brasantha, and two of our panic species, one of which Gatton panic is an important pasture variety, and indeed green panic is considerably less susceptible than some of the more susceptible species. In good news for those who are planting forage crops or mixed pastures, legumes and brassicas are not affected. So if you're planting legume or brassica fodder, they are not affected. But sorghum is, and this is a forage sorghum that we found mealybugs causing quite significant dieback. It's the only one reported so far. Again, that doesn't mean it's not out there, but we've identified and confirmed that it's the new variant of Heliococcus somervillei in forage sorghum. Before death comes, you do get this red and yellow streaking, depending on the variety. It can be almost a deep purple or a reddish streak on bufflegrass. And we can induce these symptoms in grasses by infesting them with the mealybugs. And we've done quite a bit of sort of Cox postulates type work where we've infested grasses and then recovered the mealybug and checked that it is the causal agent. And the end point is this death of grass. And this is really what a lot of graziers notice and get very upset about, is this grey ashen crumbling effect that it causes in the grass where the grass dies. And the grass pulls out of the ground. The roots are often stripped of all the outer cortex and just left with the inner cortex, sorry, of the root system. So it's very devastating on a very large scale. But those earlier symptoms are the things that you need to be looking for in the pastures because there is still time. There's no good news on thresholds either. This is some analysis that Robert Spencer's done in our group of 2 years of field sampling across 27 sites across Queensland using quantitative sampling with quadrats and transects. Some of them repeat measures sites that we monitored over about 18 months to 2 years. And basically you can see in the pastures a response to mealybug up until about 10 mealybugs. So between 1 and 10 mealybugs, you will see an increase in the purpling symptoms or yellowing symptoms on the grass. Once you get past 10 mealybugs, that's about as much as the plant can tolerate, and we don't see any response. In other words, there isn't really a threshold for this thing at all. It's very low, and it's really a matter of presence or absence. And we have got more work on thresholds from field trials and things if anybody is interested in this. But the news is consistently that it takes a very small number of mealybugs to have an impact on even large stools of grasses. How does it kill the grass? Well, we've done transcriptomics on this with Mel Monroe, and Ina is doing more at the moment because she's looking at resistance mechanisms. Basically, the mealybug is a sucking pest. It feeds on the sap. The first thing it does is inject saliva. And we can see suppression of the plant defenses and the uncoupling of the jasmonic acid and salicylic acid crosstalk within 24 to 72 hours of the mealybug starting to feed. The plant may live, but it's unproductive. And what this uncoupling of the JASA pathways does, and also suppression of many antifungal genes in the plant genome, is leave the plant open to secondary infections with things like fusarium. We often find fusarium taxa associated with dead and sick grasses. It's usually that we rarely find the same variety of fusarium twice. So there's lots of different opportunistic fusarium infections that are taking advantage and becoming necrotrophic in these pastures. And we think it's those that cause this ashen grey death of grass. the good news is that, oh, sorry, just to say how fast can they kill? This is actually an embedded video, which I won't show you in the interest of time, but you'll find the first symptoms within 4 days of infestation. And all treated plants will have extensive symptoms by 21 days, and mortality can occur within 30 days, but it can take up to 3 months. And we think that's something to do with the load of microorganisms, and particularly facultative necrotrophs like Fusarium in the soil. So there is time to do something about the management if you feel like you are in a farming situation in which you are able to take action. And it is really important to know the biology of this pest, excuse me, in order to understand how to begin to manage it. And this is everything's upside down in Australia, including the seasons. So our winter basically here is from May to September. And one of the big differences from Somerville's original paper is that the mealybug goes underground in the winter. And you can see this is an adult female with young in the soil. And then in the spring, they begin to emerge. And then through the feeding season, basically from the spring, which is October, through summer, which is December, January, and February, and into what we call the fall in March, April. So upside down from you guys, you'd have to turn the seasons around. This would be heading into your sort of September, October period here. They begin to disperse quite widely in the landscape. The mated females disperse. They're often found under thatch or in soil, and go into this winter breeding phase. Now, they are predominantly sexually reproductive, especially in the summer, but we have this evidence, and I think we've once seen it in the laboratory, where they may be asexually reproductive. And we think that in winter, they may be asexually reproductive underground. But they are sexually reproductive. It's a close to 50-50 male to female sex ratio. The males are winged and surprisingly mobile. This is a male and female mating on a leaf here. And the females turn pink after mating. So you can find the mated pink females down here in this bottom corner quite easily. They stop feeding when they've mated, and they disperse into the environment. It's a real challenge if you're keeping them in a laboratory culture, because they will walk quite considerable distances. And they disperse into the thatch below in the stools of the pastures. They will go into cracks in the soil, and they can persist over the winter. We can find corpses of females in the soil and in the thatch in late winter, and the young will erupt out of those corpses in the spring, so that they can overwinter very successfully, which has got serious implications for their potential climatic distribution. They can also do this during drought. So if you get a drought period, they will come down off the green leaf and disperse into the drier, into the more humid and safe environments in the deep thatch and in soil and under cowpats, and under logs. So you can find them in quite a bit, and they are highly mobile. They walk around a lot. They will drop off the plants very easily if you touch the plants. And the very small early instars are highly mobile on the wind. And in fact, if you breed them in a laboratory and you open the cage that they're in and you breathe out, they will drift off in the breeze from your breath. So they're able to move very readily and over long distances on wind fronts. And we think that's how they spread over these large distances. And I'm not surprised to find your outbreaks in the states that you've listed, because they will almost certainly be moved around on storm fronts. They do move rapidly into these different refugia in different seasons, and we have found them up to close to 1 meter underground in cracks in the soil. And this is my life, is living in trenches in paddocks in winter. I said I was a field biologist. I meant it. Our seasonal biology, and again, please remember that this is backwards. So summer is November to March. You can see, we can see low-level breeding populations with low-level symptoms over the winter period, with a rapid increase in the number of small and medium insects over the summer period from October onwards, with a peak around Christmas and February, March, which is the peak of our summer. So that would be sort of June, July, August for you. And then a rapid decline, usually from about March onwards. And rainfall will have a significant impact in the year that this data refers to. There was very heavy rainfall in March. One weekend we had nearly 1 meter of rain. And they are very susceptible to rain. They do fall off the plants very quickly and easily, and they will drown on the ground. But they seem to persist over cold and dry winters and drought periods quite readily. Because that's those seasons are when the mealybugs are active, is that summer period, and they're largely underground or adult females which aren't feeding over winter. You really need to target your management to those susceptible above ground or feeding populations. We did quite a lot of insecticide trials, both in screen houses and in trials, with different rates of systemic insecticides like imidacloprid and spirotetramat. And there are some new products coming out. The insecticides are very effective, but again are short term. You can see in this trial there's a hard stop just about to the right of that flag where we sprayed up to, and you can see the grasses are able to recover. Those grasses were heavily infested. If you spray them with insecticide before they die, they can recover. So we don't think that this is a pathogen that the mealybug is vectoring. We think it is an effect of something that the mealybug is producing that weakens the plant's immune system and leaves it open to secondary infections which kill it. But if you are able to manage the population, the grasses are often able to recover. The problem with insecticides is that reinfestation is fairly quick. As soon as any of that residual chemistry is gone, the mealybugs can very quickly reinvade those treated grasses. And we don't recommend insecticides for any widespread use. They're great for incursion management if you've got a small population. I'm sure your turfgrass industries will be using systemic insecticides to manage this, as they do with just about every other pest that attacks turfgrass. You need to target those emerging spring populations. There's no point attacking them when they're underground, they're not feeding. And you can't get the spray into them. You can't attack the adult females with insecticide because they're not feeding on the plants, although you could use a contact insecticide or something like a synthetic pyrethroid. But again, that has significant knock-ons for your integrated pest management and the maintenance of beneficials. So we really don't recommend that. Again, dry grass doesn't take up systemic insecticides through the leaf, and you don't apply them to soil. So it's not really viable for large areas. But in Australia, we do have this widespread use of imidacloprid in sugarcane, and we think that's why we're not seeing the big effects in sugarcane that you're seeing in your low imidacloprid use sugar production. What we do see, though, and we have seen in the field, is that there is a great deal of difference in susceptibility of different pasture varieties. And so we've done quite a bit of work on screening these and beginning to understand the mechanisms of different types of resistance and tolerance in pasture varieties. These are the results of infestation assays in the glasshouses where they're fully replicated, and we included a standard of American buffle chancrus in each assay with, I think it was 6 replicates of each variety in each trial. And we can calculate a sort of relative susceptibility based on the number of mealybugs that we recover after a couple of months. So American buffle easily supports quite large populations of mealybugs, and there are some varieties such as Lolium perenne annual ryegrass and some of the bottle grasses and some of the digit grasses on which mealybugs will increase even more, or did at least in our assays. But a large number of grasses actually support much smaller populations. And if you go part of the way down this list, you'll find that perennial ryegrass here, which has the epicloe endophyte, a fungal endophyte AR37, which was developed by Linda Johnson and Alison Poppe and several others in New Zealand against their pasture mealybug, Balanococcus poe. And we find that Lolium perenne with that AR37 endophyte on board is really quite resistant to pasture mealybug and supports very low levels of reproduction. And with the AR1 endophyte, another variant of epicloe, they are highly resistant. The mealybugs do not survive, and you only get very low numbers of mealybugs growing and surviving on perennial ryegrass with these endophytes on board. But we also find several other varieties that are less suitable as hosts and in which the mealybug populations don't increase so much. And particularly importantly, in this case, are these panic grasses, Gatton panic and Mekong brysantha, and also this curly Mitchell grass, because it is such an important native grassland in Australia. Ina Hernandez Europa is doing her PhD now. She did an MPhil on this and has gone on to do her PhD, as well as leading the writing and research on the systematics paper, which was based on her MPhil. She's now looking at the different mechanisms of resistance and tolerance and different ways of screening. These figures are based on laboratory screening assays with caged insects on grass leaves. And again, comparing them to a standard, which is American buffle. And at the top here, you can see these varieties highlighted in dark green have a very low survival to adulthood, less than 10%, and in some cases, less than around 1 or 2% only of the mealybugs will actually survive to adulthood on those varieties, and all of them have these variants of this epicloe endophyte on board and are sold as commercial seeds in Australia. And they're good for more temperate areas and cooler areas and coastal areas with higher rainfall, these fescues and ryegrasses, but not so much for the dry inland arid pasture areas that is actually the majority of the beef production area in Queensland. Green panic down here sits somewhere between these highly toxic endophyte carrying grasses and the next batch of grasses, which include Mekong brysantha and Gatton panic, which again have very low levels of survival relative to American buffle. And then at the bottom, the more susceptible ones, which include rice and sorghum, American buffle, and some of the other brachiareas, on which we get quite good survival with around 50% of the mealybugs making it through to adult stage. And these are adult females. We exclude the males from these assays because they pupate as third instars, so it messes up your data. If we then look at the female growth index, you will also see this is a calculation based on the proportion of females that develop to adulthood and their mean time to develop to adult. So these assays are conducted over about 2 weeks with regular monitoring throughout that time. We see again a similar pattern in the rate of development of those females to adulthood. And again, very slow development, even amongst the ones that survive in the grasses carrying endophytes. And in these sort of intermediate tolerant varieties, which are very important pasture species for drier grassland areas. What we do find is that where you get lower survival, you also get slower development. And that's a highly significant correlation. Where you get greater survival, you get greater symptoms on the grasses. And where you get this a higher female growth index, you get both a greater speed of development and percentage of symptoms developing on those leaves. So we're able to measure tolerance and resistance by a number of different metrics in this process. What Ina is going on to look at now is the using transcriptomics and whole genome sequencing. She's looking at the relationship between the new variant mealybug and its symbiont, and the tolerant and less tolerant varieties and the differences in the transcriptome to try to work out what it is about this new variant that is particularly virulent and what it is in some of these. I mean, obviously the endophyte carrying ryegrasses and fescues are carrying a toxin producing endophyte, fungal endophyte. But these other intermediate grasses don't, or not, as far as we can tell, don't have any fungal endophytes in the seed-borne endophytes. And so starting to look at what may be the differences in tolerance and resistance in those different varieties. We don't just do paddock work. We have a number of management trials and demonstration sites going on, funded by Meat and Livestock Australia. And we've been working with AHR, an Australian agronomy company, over several years, and we have a very healthy collaboration with them, looking at different trials. And when it comes to management, I want to talk to you. Management is difficult and challenging. What we're finding in our demonstration sites is that you get really reduced symptoms and good yield using things like gat and panic with stylo, which is a legume, in several of the pastures, and with Mekong brysantha, with butterfly pea. And those two grasses between them fill in a large part of that gap between the coast and into the very arid areas. And this is around Banana Station. That's a very happy agronomist standing in his field of gat and panic in an area that had been absolutely devastated by mealybug and pasture dieback and had been largely American buffel prior to this. We're also working in a coastal farm that has been destroyed. Its Queensland bluegrass has been destroyed by Pasture Mealybug. And we're working with them to plant, we planted Mekong brysantha and butterfly pea on about 10 hectares, and it's doing very well. Banana Station is always an early adopter. Mark Wilson's very keen, and they took up this challenge several years ago now, establishing these pasture varieties and doing a fully integrated re-establishment of long-term pastures by testing the soil, and a lot of Australian soils are very low in phosphate, using a break crop such as sorghum or lucerne for a couple of years, and resowing with gat and panic, and then rotational grazing with slashing off of any aerial tillers to reduce aerial tillering, encourage low-level tillering, and to reduce runoff between the stools. And they've had, in fact, that paddock was the same paddock a couple of months later. Very good results with this process and excellent returns. I haven't put the data up here, but they weighed the cattle in, they weighed the cattle out, and they did the maths on what they put in and got spectacular returns on investment where they took their best land and invested in replanting. And we're seeing this again and again, that some graziers are choosing to take their best land and to establish improved pastures that they can rely on, often with legumes in the mix, because if the grass does go down, the legumes will take over instead of the weeds and still provide some level of forage, as well as providing nitrogen into the soil. We've also seen anecdotal stories of crash grazing and slashing or baling in early summer, which for us would be around December to March, which for you guys would be June to September, August, when the mealybug population is at its peak and above ground. This was anecdotal reports from graziers. Coonabar Station near Rolleston in central Queensland is a very important and large station, and they noticed that if they crash grazed, they found mealybugs, they recognized it for what it was, and they crash grazed their paddocks. They had enough stock to be able to do that in February and didn't have any further problems with mealybug in that paddock for the next 12 months. We've seen this in tram lines where somebody's put a new fence through, and you can see the yellowing on the right-hand side and left-hand side in the Billowila buffel grass there. Is showing signs of dieback, but where they'd slashed it to put that electric fence through, they didn't get the dieback in the same way. And what we think might be happening here is that by destroying the grass when the mealybugs are at their peak feeding around that peak summertime of peak breeding, we're actually removing the breeding population from the paddocks, and that they're not invading quite as quickly as we would expect. We've actually opened the canopy up so they can't go down into those deeper thatch-ridden, humid areas underneath where the females like to breed and reproduce before the young move up and feed on the green leaf at the top. We've done several trials on this, and what we found is a single consistent statistically significant effect, which is if you slash, you can see there's the control there, And then we slashed in November, which is just before Christmas, which is this late spring, and then March, which is late summer. We get consistently more green. We also did some slashing in April, and these were trials where we did, on the same site. We found considerably more green grass, asymptomatic green grass, as a proportion of the total leaf cover in the areas that we slashed around peak summer. So that would be June, July, August, and possibly into September, the beginning of September, if our summers align with yours properly. And we found this at multiple sites. And we also found the same effect at 12 months, in fact, 15 months after the period of slashing. Those effects persisted in those pastures, with still significantly more green and healthy grass in the areas that had been slashed at peak mealybug season. We couldn't actually measure the impact on the mealybugs themselves because of the seasonal differences in the mealybugs, which drove us nearly nuts, but we can find it in the degree of symptoms at multiple sites, including fully replicated trials on our own field station, up to 15 months after treatment. So we think there is something to this idea that if you've got mealybugs and you find them around early summer and midsummer, graze them or bale them if you possibly can, and cut that grass down, because at least that gives you something before your grass dies. So you're getting something off it, but also that there may be a longer term benefit in reducing the mealybug pressure at that peak breeding season. In other good news, this is Edward Bryans. He's actually in Louisiana, I think, at the moment. He's with Luke Krasseline from the Natural History Museum, and they're doing a parasitoid survey tour of Florida and through to Texas. I think they get to Texas this coming weekend. They'll be at A&M Station. They may even be in this webinar because I sent them the link. Edward is a very passionate and keen parasitoid wasp biologist. I wanted him to work on fungal populations in the soil, but he's taken to the parasitoids, and he was working first for us from 2020 to 2022, looking at parasitoid populations, and really found just a single species, which was this inserted wasp, Parectomoidella. It's a new species. It's not been previously described, and he's been working with John Noyes at the Natural History Museum in London to identify it, even through the lockdown. And it was occurring really with less than 1% parasitism. And when he did sweep net surveys of those paddocks, he found it was really abundant early in the season when there were very few of the pasture mealybug around, and had quite low parasitism rates and low sweep net occurrence later in the summer. Switched to 2024, 25, and Edward's been monitoring fixed sites on a regular basis. Through this season, he's finding now 4 different taxa emerging from the new variant of Heliococcus somervillei, with 4.25% parasitism of the mealybugs collected. Basically, he's collected over 1,000 mealybugs at 8 different sites between October and June, which again is our summer into winter. About 50 of those formed the mummies, some of which have the black spots that have those marks that indicate parasitism. About 84 parasitoid wasps emerged of 4 species, the Parectomoidella, a Ropus species, which was quite abundant, a small number of Anagyrus species, which we haven't identified which species of Anagyrus yet, and an unknown specimen, which he hasn't yet identified. And we're really excited by these because what we think we're seeing is parasitoids, particularly these anagyrids, chalcid species of wasps that are moving from other hosts and adapting to the abundant resource for them, which is this pasture mealybug population, this new invasive variant. And this, over these two years of seasonal sampling at these 8 sites, he's also found a strong correlation now between abundance of mealybugs on the bottom at those sites, which was quantified with quadrats and transects, and the number of parasitoids showing up in sweep nets. So we're getting peak populations now of those parasitoids, particularly the Parectomoidella and the Anagyrus at the peak mealybug pasture mealybug population. So we think that those mealybugs, those parasitoids have come from other mealybugs and have adapted to this resource that is the pasture mealybug that's spreading. And he's very excited by this Ropus species because it's a gregarious species. We're not sure if it's polyembryonic or not, but it's certainly producing several parasitoids per mealybug wasp. And it also appears to be parasitizing without the black mark, Erin. So it's parasitizing early stages of the mealybug and producing several wasps per mealybug, which is great for biocontrol. So we're seeing an increase and becoming more seasonally aligned with our pest, which is all good news. But it's slow. We're also interested, and I am coming to an end, in what happens not just with these seed-borne endophytes of epicloe in the perennial ryegrass and fescues grasses, but also in what's happening in the soil communities of rhizospheric endophytes, fungi that colonize the roots of these grasses, and particularly in species such as Trichoderma, which you may be familiar with. Robert Spence is doing his PhD. He is looking at the interactions of species that we found in the soil associated with pastures that appeared to show significant tolerance or recovery from pasture dieback. And we actually observed the spread of circles of healthy grass in some of the pastures that had been heavily affected when we were doing repeat monitoring, and the eventual recovery of the entire pasture. And we found that that's due to several different taxa of fungal, of these rhizospheric fungi that exist in the soil. And what Robert, having done that analysis of 22 sites with repeat sampling through using quadrats and transects to tie in mealybug damage with the fungal populations in the soil. And he's now looking at some of the mechanisms by which particular fungi, and not just single isolates, but interactions in those field systems, are leading to greater resilience in even susceptible varieties of pasture grasses like bluegrass. And you can see this is just some plates that we use for screening. We also use them for third-year prac classes in microbiology, where you can see interference between different isolates. And in this corner, in the center here, Fusarium oxysporum, you can see penicillium here leading to a clearing zone where the Fusarium isn't able to spread. And down in this corner, Fusarium fetens fighting off Penicillium citrinum, and nothing gets past Penicillium citrinum in a field, but also Purpuricillium lalacinum over here, and Clonostachys here. You can see that there are interactions and clear zones where things like Fusarium aren't able to spread, and therefore may not. We think we see this in the pastures where those secondary infections from things like Fusarium are actually being resisted by some of the microbial communities or fungal communities in the soil. And in modeling 2 years of field sampling data, Robert's been able to show that where you get greater Clonostachys or Trichoderma in the soil as colony-forming units, this was all done by isolation, not just by in silico studies, you get fewer mealybugs, but there's no evidence of synergy between those two. Whereas where you get Trichoderma in the field with Penicillium, there's a strong synergistic effect where you're getting interactions between some of these beneficial fungi that result in much more resilient soils. So soil health actually does matter. It's a real thing. It's not just a fad. And we can see it with these microbial communities that they are able to provide some level of protection to pasture grasses that may otherwise be affected. And we also have some insights into the kinds of factors that lead to an increase in those fungi in those fields. So just to conclude, it's a pest. We can beat it. It's not the bogeyman. It's not an unknown nightmare that's spreading. It's a very serious pest, but it is a pest, and we can beat it. We're building up here towards an integrated pest management strategy of monitoring to know the early symptoms, to learn to find the mealybugs, to be proactive and seasonal, and understand when management can be affected and when it won't have any impact on the pests, so that you're not needlessly treating pastures and crops that might be effective. That grazing, baling, and mulching may reduce thatch in summer breeding seasons, but if nothing else, it lets you use your pasture before it goes down to the mealybug. The break and forage crops are useful if you're going to replant with tolerant grasses and varieties, and there will be tolerant grasses and varieties in your seed catalogues now. This is not research for 10 years from now. This is things that we believe that you'll be able to find in your varieties as well, to use for those graziers who are able to improve their pastures. And we recognize that this isn't suitable for everybody, but it's the kinds of things that we find do have some sort of management. It's the same with grazing and baling. It doesn't suit everybody. Not everybody has the livestock numbers to be able to rapidly graze down a patch, or doesn't have the kind of landscape or equipment access to be able to bale. But it is something that does have a beneficial effect. You can improve your pastures with legumes and brassicas. And we've seen a link between increasing legume diversity in fields and increases in some of these beneficial fungi that are providing some sort of resilience. So in addition to providing nitrogen to the paddock, and in addition to providing fodder when the grasses are being affected by dieback, and in addition to keeping down weed populations, including legumes and possibly brassicas in your paddocks, may also help to improve the soil communities and the microbiome, these fungal communities that may be providing some level of resilience. And we think that one of the baseload measures for Australia is to improve recovery of pastures by including soil nutrition, and particularly by incorporating phosphate, in which Australian soils are very low. I'd like to thank Meat and Livestock Australia, who funded us for the last 8 years, and our funding will come to an end next year. The Australian government and Queensland University of Technology, who provided the PhD scholarships, and many students who participated in this research. The excellent graduate research assistants who've led this work, really, Darcy Patrick, Jen Dixon, and Boyd Tarlington and Sean Martin, who both left now but did a great job. Two postdocs, Naimal Islam, who worked on soil endophytes and these rhizospheric communities, and Mel Monroe, who did the original transcriptome work, and both did excellent work. And my co-CI, Brett Williams, who's a plant pathologist and co-supervisor of the students, and an excellent partner to work with. The three students who are doing the majority of the work at the moment, Ina Hernandez-Europa, Edward Bryans, and Robert Spence, and particularly to our professional collaborators, Mark Schutze at Biosecurity Queensland, who's been invaluable in working on the morphology of the variants, Ros Merrington and Peter Gillespie at New South Wales DPI for providing collections and specimens for our work, Greg Evans at USDA, who provided us with information on the Puerto Rico specimens, and Z Ahmed at Clemson, who's been particularly helpful in looking at the Barbados population, and has also recently submitted a paper using cytochrome oxidase 1 markers to differentiate between the old and new variant. And I'm not giving anything away when I say that while you can tell the difference between the two variants, we cannot yet, even using the cytochrome oxidase 1 markers, differentiate whether they are different species or different biotypes or just different variants. They are within the taxonomic separation that they could be the same species. Personally, I think it's probably going to turn out to be two species, but I'd put money on it, but it's not a professional conclusion yet. Naomi Diplock and the farmers at the different farms that our trials have been working, Naomi's trials that we've been collaborating on for the past 6 years, and Jake O'Brien, who is an agronomist now at Syngenta, and the farmers that we've been working on our demonstration trials through Central and Southeast Queensland. Thank you very much indeed. And that's it for me. I'm very open to questions.
And we do have a lot of questions that Dr. Erin Powell is going to help us get through in just a second. I did want to remind folks that both Caroline and Erin are on for questions, but so is Hannah Penn. And Hannah Penn is from USDA ARS. So if you have any questions for Hannah, Hannah is also here to answer your questions.
All right, sounds good. We'll start at the top. I'm going to go ahead and just have Carrie answer all of them because I think that they're more interested in your perspective from the Australian side of things because
It's the
some of
home team, is the home side disadvantage, yeah.
the first question is where was the first find of the new variant and any idea about its origin?
The first find was that 2016 population where we know that it was, in a mixed population with the old variant that Desley identified from a sample that was submitted in 2016 in Australia. And that's the first record of it as a new variant. But we know from the damage that's been occurring across Australia and the rarity of finding those old variants, that it's probably been there since at least 2012 and possibly back another 10 or 15 years before even that. Where's it come from? We have no way of knowing. They're more from Central Asia. Heliococcus as a rule are more from that sort of Central Asia area. So, based on what we know, we don't know how it's got from sort of Pakistan area or Central Asia down into Australia. But if you want a betting scenario, my gut feeling is it's been following pasture grasses and sugarcane down through Asia, Indonesia, and New Guinea into Australia, and it's been moving on storm fronts in, and that's how it got to New Caledonia in about 2000, and it was probably in Australia then. How has it got to America? I don't know, but these things can definitely move on the wind. So we know that very small insects that can move on the wind and are very tolerant to quite harsh environments can travel extremely long distances on storm fronts.
All right. I'm curious, do you know about the New Caledonia specimens? Were there any specimens preserved in
They're
Australia?
in Paris.
Okay. Yeah.
There's your excuse. We've not been able to get to them. Paris won't share specimens with Australia since our quarantine people destroyed some one time when they weren't properly labeled. So we haven't been able to get hold of them. But, we keep threatening that somebody's going to have a holiday and go and have a look at them. We believe it's the old one, but we don't know for sure. And it was never properly described. So first in, best dressed.
The next question is, have you had any observations on corn, field, and or sweet corn?
No, and we haven't seen it on sweet corn. Great. We have found it on sorghum, so we wouldn't be surprised if it jumped. But so far, nothing seen on our sweet corn. But again, given our insecticide use, that's not surprising.
And to answer for the US, there are reports on corn in
Yeah.
Texas. I haven't actually seen specimens from corn myself, but there are pictures of corn being damaged of like the kind of like a lot, like the front of the field only when it's next to a heavily infested pasture. So time will
Yep.
tell, but definitely sorghum as a host in Texas at this point as well.
Yep.
So, next question, has the status of vectoring already been tested as is there evidence of species vectoring viruses in the same genus?
No, there isn't. And we see no patterns. We did actually look at RNA signatures in plants to look for viruses. And there are always viruses in grasses. There are lots of viruses all the time in grasses and sugarcane. What we weren't seeing was any consistent patterns of a virus in affected grasses. So we see no evidence of a virus in the mealybugs, and we see no evidence of it being a vector. What's more, because when we spray insecticides and we kill the mealybug, the grasses can recover. If they were vectoring a virus, that virus would have been introduced and the plant would continue to die. So because we can actually save the grasses, it goes beyond Cox postures. This is the house effect. If you treat it and the treatment works, that's probably what was causing it. So it's like, you kill the mealybugs, the grasses will recover. That's not vectors.
Next question, by the time you see 10 mealybugs in the canopy, how many are on the roots?
Do you know that's really seasonal? We don't find very many on the roots in summer. They're nearly all up in the canopy or down in the thatch, but they're not down in the root zone. They really move into the root zone when it's cold and dry. So in summer, we don't find them in the roots at all. It's really an overwintering thing.
We have another roots question, but I'm going to go in order so that Kayla can keep up with making them pop up. How do you survey for mealybugs? Is it looking at foliar tissue? To what degree do they feed on the roots versus other tissues?
So when we're doing it for research purposes, what most graziers do is they go out and have a look. We recommend using a hand lens because I can't see them anymore, and a lot of people can't see the very small ones. So use a jeweler's lens to look at them, preferably one that has a built-in light. They're pretty cheap to get on Alibaba. But we do a transect, 50-meter transect with 10 quadrat points. We take a 25-centimeter area, we dig it up down to a depth of about a shovel. So about 20 centimeters, and we put it onto a tarp at the side, and then we do a 10-minute whole destructive sample of the soil, the thatch, the plant itself, and the leaves. And we start at the top and we work down the whole plant and into the root zone. And we do it for 10 minutes. And we found that comparing whole counts from destructive sampling and 10-minute sampling, you get most of them in 10 minutes. So we can actually see the distribution between the soil and the plant. So we know about that seasonal biology. But yeah, we do 10 transect points on a 50-meter, 10 quadrats on a 20-meter, on a 50-meter transect point. Gives us a pretty good idea. You can usually see the symptoms, so we know what we're trying to find. And we can, we've actually got a short video of how to do that. And I'm going to make that available to Kayla and Kennedy after this meeting so that they can make it available as part of the video upload, showing how to do it. We had fun making that.
The next question is, how did you control mealybugs in quarantine, and what are their major dispersal routes?
We don't do quarantine work with this, so that's more a question for you guys. But dispersal, these things will blow in the wind. They will crawl. They will crawl quite significant differences. They can hitchhike on clothes and vehicles. They can probably hitchhike on cattle. You get very high infestation numbers. They're all over the cattle, clothes, vehicles. We're pretty sure that they can move in hay. Nobody's really done it, but I got 3 rounds of hay delivered to my property to feed horses, and I had patches of mealybugs spreading out from them about 3 weeks later, which I sprayed out with imidacloprid. So it probably can move on hay, but seriously, the main route of dispersal is going to be wind and storm fronts. And I'm not prepared to be too precious about hay and livestock movements beyond sensible biosecurity practice for your farms. I think that's what you do. But I wouldn't take it to extremes. And we certainly can't afford to be picky about hay supplies in Australia. We've faced too much drought.
So as far as the quarantine in the US, speaking to areas that don't have pasture mealybug yet, keeping in at least a BSL1 with a lot of moats, a lot of growth chamber, a lot of sticky boards at every entrance. And I've also heard about people using dark lab coats so that they can see potentially the crawlers on their coats because they're not going to show up on a white coat. So, but definitely a very, very tricky insect to potentially keep in quarantine because they're almost mite-sized as first stars.
So small. The males are highly flighted and in fact will be attracted to lights at night. So sticky boards and light traps might well find the males at night because they will coat farmhouse windows if there's a big infestation going on.
Wow.
Yeah.
I've done some measurements of the first instar crawlers, and we're looking at like potentially only 100 microns wide. So think
that's
about
right.
that for the mesh that would be required to keep these things under control.
Yeah, we can't even keep them in bug dorms. They will get out.
So the next question is kind of about plant immunity. I think it's more of a comment, but I think that you have, spoke a lot about plant immunity and plant health and the importance of mineral cycling in the soil. I don't know if you want to speak to that a little bit more there.
A lot of temperate grasses have endophytes in them. There's much less known about tropical endophytes, though I know that Linda Johnson in New Zealand is expanding her work in that area. The soil health definitely has an impact, and we can see from one side of the road to the other where there have been differences in long-term management of soil. And in very healthy soils with a healthy microbiome, the grasses will still have one or two mini bugs on them, but they're basically able to tolerate it. Whereas in the paddock next door, the grass will die. And so good healthy pastures is really important. We're not quite, we know what the reason for a lot of those ryegrasses and fescues, perennial ryegrasses with endophytes on board, is that the endophytes actually produce alkaloids that are toxic to the mealybug. They can also produce alkaloids that cause staggers in livestock, but they've been selected to have low levels of alkaloid that will affect them, that will fight off the mealybug, but don't affect livestock and don't cause staggers. And the New Zealanders did a lot of work on that work. So that those are commercially available, and DLF Seeds, which is one of the biggest biggest seed producer in the Southern Hemisphere, do sell those and supply those varieties, which are good for the environments in which fescues and perennial ryegrass are good. The other ones, it's not quite so clear, and it's not even clear because whether it's just down to things like hairiness of leaf. It doesn't appear to be. There appears to be some mechanism by which other grasses are more tolerant. And all I can say is Ina's doing this work, and we'll do it carefully. And as soon as we know anything about those mechanisms, we will let you know. But we are seeing, we don't think it's due to microbes in the plants. We think it's something about the plant's response to the mealybug itself and how they actually cause this disruption in the plant's own JASA pathways. And if anybody's wondering why a mealybug would do that, why evolution would, why they would evolve the capacity to cause such mayhem in a plant's transcriptome, it's that they're inducing a stress response, and stressed plants mobilize sugar and nitrogen, and that's lunchtime to a mealybug. That's all they're doing. The fact that it leaves the grass wide open to death and destruction afterwards is not their problem.
All right, the next question is, Pasture Mealybug has been detected in French Polynesia in 1998 and disappeared by itself 5 years later. It's an island, but has this hypothesis been considered for the US?
Well, I mean, I'll tell you, we're seeing slow but definitely increasing levels of parasitism in parasitoids in our mealybug. But oh boy, it's slow. And I'm really optimistic that the anagirids are able to adapt and exploit them. There's potential there for biocontrol, at least on a small scale. But, do we know what happened in Barbados? Nobody's following up, as far as I know, with that population there. But that was the old variant. I don't know whether it will happen. We're certainly hopeful that in the long run that there will be a more stable equilibrium. But at the minute, we're still, even 10 years, 15 years later, we're still in the invasion and spread phase of this thing, and 10 or 15 years is a long time to wait for biocontrol, and it's a big island.
Yeah, I kind of say the same thing, where a lot of times we see that there's like huge explosions of scale insects and other invasives, and then eventually the natural enemies catch up to them. And certainly that could be what happened in New Caledonia, although I don't think they actually said for sure that it was like a wasp that had, taken it down. It was just like it just spontaneously disappeared. So
yeah, they speculated it was due to natural enemies, but we tried contacting them to ask them which natural enemies, and they didn't respond. So we just don't know.
Okay, the next question is, how do we effectively control the new insect pest? Do we have a system or database that one can look into irrespective of the location to get solutions? I think the question is kind of, I think that the answer will be that you are working on publishing some of these results, and you and a lot of other people have a lot of open access literature available online that is accessible to anyone in the world.
That's right. We put it up as soon as we did, even before we were ready to publish. I mean, we have more to publish than we have time, because we wanted to get that information out there. I've given you a quick summary of the best that we have, which are basically varieties, seasonal management, pesticides if you're in that kind of turf grass or cropping industry where pesticides are relevant, but not really for pastures, and resistant varieties and soil health. It's possible that there may be some effects from inoculants, microbial inoculants. We haven't seen that yet. We see it more with natural establishment of, and it's more than one microorganism to really get the effect. But we're sure that somebody, and we're looking at it as well, which varieties of fungi might be used as biocontrols. And I believe that it's worth having a look at some of the commercial anagirids, which we weren't able to get. So there's Anagyrus in Australia that was released as a biocontrol, but nobody supplies it anymore. I know Cloppert still make it, still produce it. It's quite possible that they will adapt as well. But there's lots of things that will add. There's no silver bullets that are going to fix it unless you're a sugarcane grower and you're going to spray imidacloprid. But if you're out in the field and it's pastures, there's small things you can do that will take 10 and 20% effect and add up to a management program rather than a single cure.
Okay, and the next question is, it should be a global issue, not a national issue, as international trade
Agreed.
continues to introduce new pests to other countries. And again, totally agree. And we're working on getting as many resources out. And like this webinar, is an international collaboration with Australia and the US to try to get everybody together and
And I
distribute.
was at a meeting with CABI, CAB International, 2 weeks ago and raised it with the representatives there that this is a serious issue and it's got repercussions for South America, North and South Africa, and Europe as well.
Okay, the next one, here in Texas, we typically have bimodal peaks of rainfall in late spring, early summer at about 90 degrees or 32 Celsius. And again in early mid-fall when temps are still regularly between 26 and 32 degrees Celsius. Could this result in 2 peaks of mealybug activity if the temperature stays warm enough?
Look, we found a lot more mealybugs around in May, June this year than we've found previously as well. And in years when we've had very heavy rainfall in late summer, we have that bimodality as well. We have a storm season in spring, and then we have a wet period around Easter time. That doesn't mean anything to you in late summer. And that the peak breeding season really is from late spring through until late summer. And they build up quite rapidly over that time. You get a rapid build up through the first few months of spring and summer. But then they do die back quite quickly. And if you do get some cold weather, and you do in Texas, Queensland and Texas are very comparable in terms of climate. So, you might see knockdown when you get rain, but they will certainly, I would expect them to die back quite significantly in winter and then re-emerge in spring. I hope that answered that question.
Yeah, I guess it'll just be a time will tell on how many generations we get through. And even within Florida, our climatic, distribution across South to North Florida is so different that we might see very different things. This person says that I'm in Central Texas and have strong pasture mealybug pressure. I'm looking to work with them on some biological and mineral treatments to shift the sap makeup of my grassland acres to see if we can apply their knowledge with sap analysis to a grassland setting. They have extremely positive results with cropland and orchard system sap sucker pressure.
Yep. I'm not quite sure what you're suggesting there. We always recommend phosphate soil test and address soil nutrition so that what you do have can recover. We strongly recommend, where possible, to incorporate diverse pastures with different grass varieties and legumes and brassicas as forage in the mix, or establishing forage areas of intensively improved pasture on your best land. Invest with the best. It's where you get the best returns. Don't worry too much about marginal land. So improving pasture varieties, improving soil health, particularly phosphate. Mealybugs love nitrogen, so be careful about just applying more nitrogen. That's what mealybugs do. They like sugar and nitrogen. So be careful. What your cattle respond to is what the mealybugs will also respond to. And yeah, generally watching out for that seasonal peak from just approaching midsummer, which would be in about June, your time, through summer. And if you are seeing large mealybug populations, graze it if you can. Bale it if you can. Keep it for the fodder it's worth. And we don't recommend slashing because it reduces all your biomass. But if you've got nothing else, then at least get that canopy down, and it might have some effect. But I also know that people like to lock up their pastures for later and see them through the winter, so that's a bit difficult. But baling would be a good option. I'm not sure I answered the question at that point. I hope I did.
Yeah, I think just still a lot to learn with the seasonality of everything. And
Yeah, there is.
yeah, I know that a lot of our producers are trying to grow like standing hay for the winter, which is definitely a challenge with this right now. So
it really is. Standing hay is a serious challenge with this, and I'm much more fond of baling or silage if you can do it.
Mhm. Next question is, have you found pasture mealybug on flooded rice?
No, not much rice in Australia actually, and it's mostly up the top end where we don't yet have the pasture mealybug, so we haven't seen it. But flooding would be very antithetical. The mealybugs wouldn't like flooding, so
Yeah, that's kind of been a question because of the, the Indian paper that had it on potted rice. So then rice shows up as a host everywhere. And I've gotten that question a lot. Is like, well, are they going to survive on flooded rice? And I think that some of the later questions kind of allude to this is, do you think that flooding sugarcane or some of our pasture grasses that can tolerate flooding like limpo grass, do you think that that's a good strategy?
do you know, I don't know, but we see certainly where we get very heavy rain events, it really knocks the mealybugs back. Both the impact of the rain, but also the ground level flooding. I don't know how long you'd get that effect for. And we don't use flooding. I mean, even in rice in Australia, it's dryland rice. It's not done with flooding rice. So the answer is I don't know. But we do know from very heavy rain and flooding events that the mealybugs will reinvade, but they don't survive flooding.
Were there any natural enemies recorded in these field trial sites in Australia? That question might have been before you got to the wasp.
Yeah, we're up to a guaranteed 4 different parasitoids, and there are also things like cryptolemus and predators, but they tend to be very patchy and late to the party when it comes to getting on top of the mealybug. So, but the parasitoids we're quite hopeful will actually start to increase, and I'm very happy that Edward's over working with your guys from the Smithsonian at the moment to start to look at that. That's really good.
Mhm. Looking at the survival rate in your resistance and tolerance slide, it appears that cool season C3 grasses are less susceptible than C4 warm season grasses. Is there a correlation in stem to leaf abundance on species and susceptibility? It appears
Oh,
stemmier plants have a higher infestation rates and impact than leafier plants with smaller stems, potentially from anecdotal observations.
I mean, Mekong brasantha is highly resistant. It's huge, and it's a big tropical grass, and it has massive leaves, and it's very tolerant for some reason. Um, when we actually look in the laboratory, we don't find that so easy, that C3, C4 temperate tropical thing to work. I mean, the really resistant ones are the fescues and the ryegrasses, but only when they've got endophytes on board. It's not because of the grass. If you get the original varieties without the endophytes, they're quite susceptible. But it's that big wodge of green ones at the top all have epicloe seed-borne endophytes embedded in those varieties. And if you want to, I'll put that slide back up if it will go, just to emphasize that point. There it is. These ones, all these ones at the top here, perennial rye with HE, AR37, Max P. There's Soteria. Max P and AR1. Those are all endophytes apart from in the Soteria.
It's not coming up. It might be because the Q&A is up.
And because I'm sorry, and I'm not sharing screen. There's an idiot mistake.
I'm not sure if we're also potentially going to send out slides. But
yeah, I'll redo the PDFs if you're going to send these out. But you can see at the top here, all of these ryegrasses, HE, AR37, Max P, AR1, those are endophytes. So it's the fungus doing the heavy lifting in those varieties. If you take out the perennial rye without any, so it's a nil, not got any endophytes, it's much more susceptible. It's still not wholly susceptible, but it's much more susceptible. Um, but that's why they're there. Green panic, on the other hand, that's not a temperate grass. These are topical grasses that we're looking at here.
Yeah, definitely a lot to learn still, again, with our suite of grass flora that's grown here.
That's right. And you have very different varieties from us. I mean, some of them are shared, but not a lot.
The next question is, has any work with endophytes been done in sugarcane?
Not that I'm aware of.
So I know
No.
there's been a few instances in other countries dealing with plant pathogens using
Yeah.
endosymbionts in sugarcane, but nothing for insects and definitely not in the US.
Mm. Yeah, we're definitely seeing something that's not been very well explored and definitely not got quantitative data behind it. We're quite happy with the data that we've got. We've been very slow to publish because, oh boy, we've been through that data before we stick our neck out. But we're definitely seeing soil communities that are more resilient to an insect pest in real time in the field without any interventions. So we're not talking about applying inoculants. We're talking about what we see in the field ecology.
Next question, have any augmentative releases been done with parasitoids?
Not yet. We'd love somebody to try releasing Anagarrus biocontrols to see whether it works. And you're going to be the ones that do that. I know that there's somebody in Australia who's interested in doing it now, but when we contacted them a couple of years ago, they didn't have any of the commercial produced Anagarrus available. So
I'm curious, do you know of any other countries that have tried to release parasitoids?
no. I mean, we're the only big country that's had an outbreak until now. So
do you have any concerns or comments on impact on beneficial insects in areas where sugarcane has been heavily sprayed lately? So I think that
yeah.
might be a question for everyone. Anna.
Yeah, I mean, the obvious one is what are they spraying? And, if they're using synthetic pyrethroids or disruptive chemistry, then you've got a problem with knock-on effects on your biologicals. That's always been the case. We use a lot of imidacloprid, which doesn't have, especially at planting, which doesn't, as a granular formulation, which doesn't have as big an impact on beneficials. But, nobody likes spraying neonicotinoids. Yeah, it depends very much what they're spraying. But beneficial, I could supply you with tables, as I'm sure Erin and others can, about the impacts of different pesticides. It depends very much what they're spraying.
Yeah, we are monitoring that in sugarcane in the US. So far, it's been relatively minimal since we've had it only in the upper canopy since we've been doing mostly foliar applications. But now that we're doing more direct soil applications, it's going to become much more of a concern.
Yeah, but I mean, granular neonicotinoids, for all you can argue against them for other reasons, at least it's not a foliar spray and you don't tend to get drift impacts.
Next one is, I think they mean parthenogenesis, common in pasture mealybugs. Do they have the ability to switch the mode of reproduction?
We think they do. They're primarily sexually reproductive, particularly during the summer, but we've seen evidence that there may be low levels of parthenogenic reproduction in females, unmated females, because unmated females stay white. I mean, if they're reproducing when they're white, they're not mated. You can actually see them acquire the pink coloring when they're mating with the males.
So you don't think that it's like that they just turn pink when they're ready to mate?
Nope.
Okay.
Is that what you think? I mean,
Well,
I'm
we've
open to,
had some
we've
that have been
watched it.
isolated that have kind of seemed to turn pink alone.
Okay. Oh, well,
So,
that's good.
but
We've
again,
definitely
very
seen white ones.
anecdotal.
Yeah, we've seen isolated females as well, where we've removed all the males. We've had one case where they reproduced,
Yeah.
and they stayed white. But
It's
we
also,
can't. We couldn't. Typically, if we separated males and females in the early instars, they didn't reproduce in the
okay.
laboratory.
Yeah, it's very tricky because there is some evidence in the literature that potentially third instar females can be mated, almost
We
like
definitely can.
traumatically. Yeah. So like it's like you have to make sure that you're isolating from the very beginning,
That's right.
not just
Yeah.
once they're adults. Yeah.
No. No, definitely not at adult. But the males from second instar are quite easy to identify because they're much longer.
Mm.
It's just boring and you have to get your eye in. And I can't do it anymore, I'm really happy to say. But, young people can do this.
On that note, do you know where the immature males pupate for the most part there?
Um, they seem to pupate on the leaf.
Mm.
They don't seem to drop off and pupate in the soil, or not that we've observed anyway.
Okay. Have you found that stargrass is resistant to pasture mealybug?
I don't know what stargrass is. Has it got a...
I think it's a type of Bermuda.
Which
They
also doesn't actually mean very much to me. So that's probably a better question for your guys.
Yeah, I mean, I think we've had it on. Yeah, we've definitely had records on Cynodon. So it's the Cynodon lymphensis, and we do have records of it on there. We certainly don't have enough data yet to say if anything is resistant here yet. So
No. Yeah. No, we've never tested it or seen it, but it's not a species that we see particularly often.
how long can the mealybug survive in baled grass or hay? I think you've kind of touched on that.
Months, the little horrors. Yeah, they go into this sort of, I mean, even the mated female's corpse can be a nice environment in which to preserve mealybugs. They can live for months after mating. If it's cool and dry, months.
Have the impacts of fire, specifically seasonally prescribed fire on pasture mealybug, been evaluated? Also interested in prevalence of native high diversity grassland systems compared to less diverse managed pasture.
Okay, yes, we looked at burning, and it's just impossible. The problem with burning is that it does two. I mean, it will provide a short-term control. Your mealybugs will not survive burning. But when you replant with another grass, if you plant with a susceptible variety or you get a seed bank regeneration with a susceptible variety, the mealybugs will come back in. So it doesn't provide any long-term control. It just destroys what's there. Well, I'd rather bale it or graze it or plow it in. The two other things that burning does is it gasifies off a lot of your mineral. So nitrogen and phosphate. I mean, phosphate is so slow to be produced and released into the soil. And you're burning it off, you're gasifying it and putting it into the atmosphere. You're also gasifying a lot of your humic acid and your other soil components that are important parts of healthy soils, and you're cooking your microbial communities that are important to soil health. We see no upsides to burning at all, except as you can do an awful lot of damage across a big area, but as soon as your grass comes back, it's as vulnerable to being infested by mealybug as it ever was, and possibly more so because of the damage that gets done to the soil when you do burning.
Does the pasture mealybug lay eggs directly to the environment and where, or do females give live birth where the egg hatches inside the female before releasing the young?
Mealybug reproduction's weird, isn't it, Erin? They produce what are effectively live young in a sac. And then in some species, they may actually be born live, and in others, they rapidly emerge from this waxy sac that's around them. We see it quite often that the females, this sort of waxy sheath that they're laid in, the females will actually spool it up with their legs and hold it against them. So that they form this sort of waxy cocoon with the female's body in the middle of it. And if that female dies, that mass of waxy young can stay in that position for quite some time, for months. And if you bring them into the laboratory from the fields and you put them in nice, warm, humid environment, the young will erupt out of it. So they're holding this sort of waxy filament around and gathering it to them as they're produced.
So the picture of them in the soil, it almost looks like the immatures are above the female.
Oh, that's because when you crack that, let me see, I've gone off screen sharing again, haven't I? But I was just seeing if I could find that picture of the mealybug in that one in the soil. So I'll share that one again. This one. you can see here, I can blow that up. Oops, go back up. No, I can't. But you can see all these little small, they're all round that female. It's actually like a cocoon and you can see all the waxy filaments. When you're digging in soil to find them, you often find the waxy filaments without finding the females around. So they're still producing what would be those little waxy spicules on the surface. But they're also producing it around these young, and these young were crawling away as we cracked that little cell open. They form like a little cell in the soil, and they just produce lots of young in it. And then in the spring, they'll crawl back up. We think that was at the bottom of an ant tunnel. But you'll often find them in the soil, and they're only above, and that might not even have been the way up they were in the soil. It's just the way we photographed it.
So is that this one that you would consider to potentially be parthenogenic since she's
Yeah,
not pink?
absolutely. Yeah, we would.
Okay. And there's no possibility that that's like a Balanococcus or something different.
No, we don't have Balanococcus.
Oh,
But no, it's definitely Heliococcus, and it was in the middle of a big outbreak of them over winter.
okay. Yeah, I've seen kind of the little like waxy thing that you're talking about where it's almost like the but it does seem like this species is kind of like, that quick. Like there are plenty of mealybugs that have an ovisac that you have a proper egg stage that you can watch for a
Yep.
while. And yeah, it's like a lot of the like wax morphology kind of correlates with what they do with those because if they have like, those structures to produce a proper ovisac versus not. And this one definitely has limited, like it doesn't have multilocular pores, whereas most things do. So.
Mhm. Yep.
Next question is, do the two variants share parasitoids?
We don't know enough. You've got more of the old variant than we have. Ours are so localized, we can't find enough of them. It's like gold. We keep going back to try and find them. We don't know. But because of the kinds of parasitoids we're finding, they're usually they're parasitoids that would normally, that do adapt readily to new hosts. They're not host specific. We even found a parasitoid of a mealybug that's only just recently arrived in Australia, and we found one parasitoid of that mealybug. The mealybug is Vibergia, and we found its parasitoid in Heliococcus, and we confirmed it was a Heliococcus as well because we couldn't believe it. So these are parasitoid wasps that will adapt to other hosts, and that's why we're optimistic about a slow but steady natural development of parasitoids adapting to the seasonality and abundance of the pasture mealybug over time.
And on the US side, we have parasitoids from both variants, but they were different things. So time will tell. It's still very early days, and old variant is also like gold here. So
Yeah.
just like, not anywhere close to being near, as virulent as the new variant. So it's going to be a lot harder to figure that out, but potentially. So we do have some preliminary stuff there.
But interesting, existing in a separate state over a long period of time in multiple environments now where we know you can get the old variant and the new variant coexisting with very different biology and different morphology. And this is what leads me to think that over time they're going to turn out to be two different taxa, two different species.
Yeah, it's certainly fascinating that the old variant is no longer pestiferous in Australia and like why that is. And it's not pestiferous here either and why that is. Are they overwintering as adults or eggs?
Adults with eggs.
Okay, so or like kind of the clump of first crawler, first instars under the female or
That's
kind
right.
of
Absolutely, yep.
The next one is a request for the link. I think the link will probably be sent to all of the attendees, but I'm not sure. Then
I'll share a few links as well to things like videos and for if there are any grazies in this meeting, things that you can look for and how to identify mealybugs. We've produced some of those outreach materials which you can have. I think Erin, I may have even sent them to you already. I don't know. I will.
Yeah, we'll make sure to kind of I think that we almost need like a central repository of everyone interested in Pasture Mealybug just to start having like a listserv, which I've kind of talked to some folks about starting a kind of world serve because there's so much demand for information right now. So Next question is, have entomopathogenic nematodes been considered or tested?
No to both, I'm afraid, and I'm perfectly happy to. I'd be very happy to see somebody give it a go. But we just don't see nematode activity in our pastures, and I swore very early on never to work on them.
And what about entomopathogenic fungus?
Well, that's what I mean, the ones we're finding are entomopathogenic, as well as it's these hypercrealium fungi and others, things like penicillium, that are multi-purpose fungi. And some of them are producing chemicals, secondary metabolites, and other products that are spreading through the plant. And they mainly live in the soil as saprophytes or as colonizing the roots as symbionts on the root. But some of them are also entomopathogenic. And you've got to remember that entomopathogenic fungi are actually saprophytes that evolved to colonize the roots of plants. But they also acquired genes that allowed them to take out nematodes and insects. So that's where they're evolved from. They're evolved from rhizospheric and saprophytic fungi rather than just being entomopathogens in their own right. But yeah, things like Purpuricillium, which is a nematode trapping fungus, and Trichoderma and Clonostachys and Bavaria, which we find very rarely in the field, but when we do, it has a big impact on, by colonizing the roots. And it's a known entomopathogen.
Do you find any association with ants in Australia?
Yeah, and I mean that's the really interesting side of mealybug ecology. The only thing I can say is that trench that I spent a morning digging in had a very deep ant crack in the soil, and the ants were moving around. And I don't know whether the ants carry the mealybugs underground or whether the mealybugs just walk down the tunnels themselves. But those cracks in the ground do. But whether we've never seen ants carrying mealybugs or farming them in any way, shape, or form. But who's to say they're not?
So we have seen fire ants, Solenopsis invicta, carrying them, I think 3 times in Louisiana, but we don't know if it was farming or not since we haven't seen any tending, even though they've been on the same plant tending other hemipterans.
Mm-hmm.
Yeah, so I've also seen a Solenopsis invicta carrying one, but again, don't know what the context was, if she was just panicking because her nest was being messed up. But what's interesting about this mealybug, though, is that their anal ring morphology is different than most mealybugs, and they have to like flip up their abdomen. I don't know if you've seen them, kind of like when they shoot
Yeah.
honeydew, they flip their abdomen up because the anal ring is actually ventral, not dorsal, which is extremely odd. And if you watch them like in a vial, you'll see that the honeydew actually will like hit the side of the vial or whatever. So they're actually like, ejecting it pretty far, which is not typical for something that gets tended by ants, that where the ant actually comes and takes it out of the anal ring directly. So it's very interesting. And we have also not seen ants tending them. I mean, you'd think that they would be just such a good source, like given how heavy the infestations are. So I think that's a very interesting question of how and why ants are or are not interacting with them.
Cool.
Is the mealybug destroyer a significant predator in Australia, or are those populations being kept suppressed by pesticide use targeting other pests?
And all of that. I mean, there's plenty of Cryptolemus, but they're not really abundant in pastures. They're very patchy. They're even produced commercially, but the cost of releasing them on the scale that would be required over the area. I mean, it's the vast areas. That map looks small, but man, that area of Australia is huge. It's like the entirety of your eastern seaboard. And and just in Queensland. So, it's not economic to really produce those. It would cost more than the land itself is worth to keep doing that. They certainly will take the mealybugs. That's no problem at all. But it's just the sort of ecology and scale of getting that many Cryptolemus across the kinds of areas, thousands and thousands of hectares synchronously infested. Cryptolemus can't touch it. Parasitoids probably could.
The next question, I'm not fully sure what it means. Which nanoparticles are used for the management of this pest in Australia?
We don't use nanoparticles for control.
How long can pasture mealybugs survive under flooding?
I
We
don't know, but they don't generally like wet conditions. I mean, they can certainly float on water, but we generally see a decline in numbers with rainfall.
Okay. That's actually another question later, I think, is have you actually tested how long they can float or how well they
No,
do? Okay.
it's a nice one. Yeah, I should get it. It's a good undergraduate project, that one. I should get someone to.
Mhm. It's lots of fun things to do with their like
Oh, there's so many things to do. Yeah.
the next question is usually we rear mealybugs on potato sprouts. Have you tried?
They won't grow on potato. They really are a grass specialist, this mealybug. There's lots of Solenopsis mealybug will grow very nicely on potato. This one won't.
Hmm. Yeah, I was thinking the same, but if you want to rear something else to kind of have food for parasitoid colonies, then
Yeah, if you're trying to rear cryptolemus or even parasitoids, then growing that mealybug on potato or tomato or pumpkins can be
mhm.
a really useful way of breeding up your natural enemies.
Did you have like a favorite grass that you had most things in colony on?
We always use American buffalo for our, yeah, we always use American buffalo as our colony plant.
Okay.
They
And
really grow well on it.
mm, is there kind of a trade-off with like how quickly the grass dies versus how well your mealybugs do and like trying to keep everyone happy?
Yeah, we have the problem is mealybugs getting into your clean grasses. So we have a glass house facility that's about 30 miles away from where we do any of our infestations or colony management work. And in fact, we have a field site which has a screen houses where we do most of our colony. And then we really try to bring them into the laboratory to combine them.
Mm-hmm.
But we have to keep our grass rearing they're actually in PC2 glasshouse containment facilities well away from anywhere. Yeah. And if mealybug gets in there, that's it. We have to start from scratch.
Is there any evidence of root feeding while pasture mealybug is in the soil? And if there is root feeding, does this cause symptoms in the canopy?
I think they were root feeding, those little cells. Sometimes we break them open, we find they're really closely associated with roots. Whether they were feeding on it before we broke it open, we can't guarantee. But we can see symptoms in grasses where there are no mealybugs on the surface and they are only in the root zone. So I'm pretty sure they do feed on roots.
Mhm. Yeah, that's been a big question here. Something we're trying to figure out how to test.
Mm.
Which I think there's some ideas floating around. So
Yeah, it's a good test to do, but they're very closely associated with roots. I mean, not all of them, they will move off, but you will often find them associated with roots, and I'm pretty sure that they do feed on them and cause symptoms. And in winter, that's how we look for the symptoms, and then we pull the grass up because they'll be down in the roots.
Mhm. Are the crawlers phototactic?
No, no, they're not. No, they're not even thigmotactic. They I mean, they often sit along the edges of the leaves. The very small ones are like line like a fringe along the edges of the leaves if there's no breath or wind. And they will blow off very quickly. It's almost like they're dispersing.
Mhm.
Like they're adapted to disperse in that first instar. They sit along the edge, up in the canopy of the plants. So right out in the open.
Yeah, I think that it would be nice if they were a little bit so that control them a little easier in quarantine, but
Yeah, that's right.
unlike other mealybugs, that might be a little easier. So the next question we just answered, do they feed on the roots or just overwinter in the soil? So we talked about that already. Alan says that for biocontainment, he was suggesting a BSL-3, similar to containing fungus. But he says later on that he had to pop off the chat, so I think he's
Okay.
not any longer. Do we have any information about pasture mealybug in nurseries or ornamental grasses?
It's been found in turf grasses and lawns. It's certainly been found in nursery grasses, unless they're using lots of insecticide.
Mhm. So to speak to that in the US, we have not found them in nurseries. However, there's like a love grass or Eragrostris. There are some species of grasses that may be, commercially cultivated for ornamentals. I mean, of course, you're talking about like turf grass is different than like a potted ornamental. But what we're actually seeing is that the Muhlenbergia does not seem to be a host so far, and I'm getting tons of false negatives, whatever. Not Pasture Mealybug. There is a specialist mealybug on muhly grass called Stomatomeryx that has also weird ducts that make weird wax tubes, and we're seeing a lot of that. But I don't personally think they're going to like muley grass because muley grass has like a really tight round ligule and not like a good like open surface area of the leaf blade for them to be on. And I just don't think they're going to like a grass like that has that really round, tight leaf. So have you seen any hosts that have a round leaf blade that
It can get down into leaf sheath.
Mm.
So, but they're generally out in the open.
Mm-hmm. Yeah. Okay. Are there any analyses taking place that look at the toxicological properties of the pasture mealybug saliva? What is in the saliva that is so toxic to the plant?
Not sure what's in the saliva at all, but Ina's taking a very careful look at all the sides of that from what, from the symbiont to the mealybug to what happens with the transcriptome. And we're not sure what it is. There's a lot of work been done by a guy called Zhang on Solenopsis mealybug, and a lot of that kind of work is the kind of thing that he's really interested in, and so we're paying attention to what he does with Solenopsis mealybug. And I can share his papers on things like jasmonic acid, salicylic acid pathway disruption, crosstalk disruption.
Mm-hmm.
I think I actually put a reference in one of the slides as well.
So soil health can be so many things, including fertility, organic matter, disturbance. Do you have a sense of what components may be most impactful?
Oh, I wish. Look, it's the healthiest ones are always the ones that have got the most organic matter in. We found one site that was just miraculous. It was an old lucerne farm that had been a pig farm before that. Now it had been converted to pasture, and the soil was like chocolate. You could grow a lamppost in that. It was just rich, and the cowpats were breaking down. There were dung beetles everywhere. It was just a light, lifted soil that was just beautiful and rich in humic content and packed full of beneficial fungi when we had plated it out. And the same soil on the other side of a tract that was owned by a different farm that had been burned and grazed and never planted with anything, and was as hard as a rock, and the cowpats were like frisbees, and it had nothing living in that soil at all. And the mealybugs destroyed every blade of grass that came up, and yet there was nothing between them. And the prevailing wind was from the heavily infested paddock into the one where on the other side of the road. It was like chalk and cheese. It's very hard to find examples like that. You're normally looking at gradations of, but they tend to be very rich. They tend to have greater pasture diversity. They tend to have more forbs. They tend to have a long history of not being burned. They're just so they're diverse. They're rich soil content, and then we take the soil away and we measure what's in it. So that's how we really do the measure of what's healthy soil. We measure the organisms, but when we're sampling it, you get a feel for knowing that you're going to find a lot of stuff in these paddocks.
Can you comment about crash grazing and if low mowing and turf could be helpful in the same way?
Yeah, I mean, where it crops up in turf grass, people swear that it mowing doesn't make any difference, but I suspect that they've got a lot of thatch in there. We honestly haven't looked at it because we just don't have time and money to be able to do it. There's so many things to look at, but you've got a huge turf grass industry, and I'm sure that this is going to be an important question for them. And look, my lawn never had it. It's that kind of level of stuff. We just don't work on turf grasses. We do see turf grasses that have overgrown get infested, but we've never seen it in a lawn as such. I don't know, that's not true, but it was a very thatchy old lawn on a farm.
Yeah, I've seen some very, very pretty turf next to some pretty infested places. So it's either the chemicals or the grass species or something is doing a pretty good job. So
yeah, we just don't know, and I'm sorry, but our turfgrass industry's never paid any attention to this. They use a lot of insecticide.
Are insecticides sprayed or added as a seed dressing?
In sugarcane, they're used as a granular formulation that's put in at planting. In most other well, I mean, seed coats for plants, but not for grasses typically. Oh no, that's not true. A lot of improved seed is sold as with imidacloprid in the seed coating, or it's put in as an additive into the seed drill. So it's put in with the seed in the mix as it goes into through the drill for planting, and that does provide some protection for a couple of months. But we've seen it with coated rose grass, for example, but eventually the mealybugs will get it as that initial application fades. We did it with spraying because that's how we were testing. We wanted the grasses infested with mealybugs before we sprayed them with insecticide. But yeah, it's used as seed coats and in drill granular formulations and added to seed applicators.
The next question we've pretty much covered is experience or advice on Ossaria or Bavaria, so entomopathogenic fungus, which we just talked about.
And other people have done trials with those and found no impacts, no benefits in the field. So we're not reporting the impacts of using inoculants, which we're looking at, but we haven't finished. But we are finding natural populations of those particular groups of fungi. We mention it in our reports, but Robert's been doing his PhD on this.
Okay. Which RNAi-based biopesticides are used for management?
No, none, not yet. No, we're not there yet.
Oh, I think I missed one. There, they change. Does the root feeding represent a big deal in the overall damage? We kind of talked about that, where it seems like it can cause symptoms.
It can, but in overall, the populations are low over winter. They don't really start breeding in numbers until they emerge in spring. So it's more a problem because of this overwintering effect. You get winter flooding and it'll wipe them out.
So because yellow, I mean, I think that my impression has been that you think that imidacloprid is kind of responsible for keeping it at such low levels in Australia. But if we're also seeing kind of like low numbers with high damage in sugarcane that isn't necessarily being treated yet, do you think that this kind of like mysterious yellow canopy syndrome like could be more root feeding than we see in pasture grasses?
I think, and this is a think, this isn't proof, this isn't, done, no experimental, but I actually think that when you see yellow canopy syndrome, because of where it occurs in the canopy, it's sort of that height where the initial ground application at planting of imidacloprid has worn off. And before they start to put on the foliar sprays, you get a band when they're susceptible to mealybugs, and that's when the symptoms will come in, and then they start spraying it, and then the sugarcane recovers. The leaves that were yellow don't recover, so you get this yellow band, but the new growth at the above it will be clean and green. And we pretty much did that in the glass house. But nobody ever paid us to work on sugar. It was a small project we did because we wanted to know. And mealybugs go off in sugarcane unless you've got Confidor in there. Thousands of the things. So we think it's that continuous application of maintained level. And when the level drops off, if the mealybug's in the right place at the right time, you'll get some yellowing come through. And then they'll spray something and then it goes away again. That's my bet. I know the sugar people will curse me for saying it, but I'm pretty sure that's what's happening.
That makes sense. And, we have not had this history of kind of planting with imidacloprid at the beginning. So it may be why we're just, kind of trying to catch up with, completely clean plants, unprotected. And yeah, if any of the sugar folks online have any more follow-up on this topic, feel free to chime in. We're going back to hay here soon.
Yeah, no, I think that's completely correct. Like in the US, we don't use imidacloprid traditionally as much as Australia does for sugarcane. So our plants were kind of sitting ducks.
Okay. If infested forage is baled, how long is PMV viable? So we talked a little bit about that already.
Yeah, it can be months under
Months.
good conditions.
Mhm. Okay. So we talked a lot about burning already, where yes, it may kill them at first, but they're just going to kind of reinfest, and then you have a lower microbiome for
And
less.
soil fertility and yeah, all the other bad side effects on the impacts on your soil are really bad.
Mhm. Um, so that was another question about prescribed fire. So we'll pass that one. We've talked about entomopathogenic nematodes and fungus. All right, if Kayla, if you can keep up with me, we're going down to Jose's now. Okay. Are you experiencing both Pasture Mealybug variant populations in your different ecoregions, or have you found a predominant or single variant in specific areas?
It's always the new variant. We found one or two individuals of the old variant in our areas. Erin, I don't know what you're seeing, but it's almost impossible to find the old variant. It's new variant wall to wall, and there's very little difference between them, even at
Mhm.
the molecular level.
Yeah, we also have extremely low numbers, probably around 5% of everything that's been slide mounted, which is probably getting close to 200 slides of adult females now. So,
5% would be off the charts for us. We're talking less than 1%. We're talking two individual out of hundreds.
yeah, yeah.
Variants.
So, ours were not necessarily like sympatric at the beginning, but, the invasion is spreading quickly. So it's hard to make any comments about that. But as far as like different ecoregions, I mean, in Florida and Texas, certainly we can't really speak to that just because the sample sizes.
No, and we the old variants that we found that were found originally were found on the Sunshine Coast, which is this um, uh, temperate but hot coastal grassland, and up in the Atherton tablelands, which is subtropical tableland area. So and a good 1,000 kilometers between them.
So we talked a bit about whether they float. They do float. We need to test how long they can float for. We talked about does sucking of the sap release a toxin into the plants that causes yellowing, and so we've talked about that. Do we see slowed or suppressed spread of infestation in cooler temps and the winter season? Seems like probably because they're underground.
Yeah, I mean ours are only as far south as North Sydney now, so we don't know how far south they're going to get. But because they've got this strong, deep overwintering capacity and this high mobility, I'm prepared to see them move quite a bit more into the temperate areas. But we just don't know yet. We're just watching them.
Have you ever seen any association with snails?
No. Oh. No.
I haven't either in all the fields that I've been to, but I guess there's a lot of snails in one of the fields in Florida that's affected.
Yeah, no, we have super snail plagues as well, but we've never really seen them at all.
Mhm. What do they do with the long wax filaments?
They break off. They shed them when they molt, they break off, they use them as protection. They're like a sort of deterrent for parasitoids. I don't know, there's lots of things that those waxy spicules, they don't really do anything with them. They just extrude them and then they seem to form a useful surface covering to protect probably from parasitoids coming in is probably what they're doing.
Did you check for pesticide residue? Because these have long periods of activity. I'm not sure what that's in reference to.
I mean, we actually did residue testing with one of our pesticides to see whether it was worth registering. And basically, once your pesticide, imidacloprid, within 3 months of most applications, the plants are vulnerable again. And sure enough, you could almost set your watch by it when the mealybugs would start to creep in from the pasture next door. So, yes, the residues can be a couple of months, but as long as there's pesticide there, then there's no mealybugs. But again, it's just you don't want to be chucking a lot of pesticide out across just the vast areas that these things get into.
Do females appear to produce only a single batch of offspring?
Yes, They really do seem to produce, in ideal conditions, they produce most of their young in the first few days, and then they stop, and they can linger, and then they might produce like one more, which really makes a mess of your life history studies. But they produce most of them within a few days.
Okay. Has anyone identified the sex pheromone used by pasture mealybug?
I was hoping somebody would ask. We have an MPhil student working on it with our synthetic biology group right at the moment. So the answer is no, not so far, but she's pretty keen to get it.
Mhm. Okay, yeah, I've heard of other people in the US that might also be interested in doing that. Would be cool to try for sure. And
Yeah.
are there any reports of field evolved resistance to imidacloprid?
No, we haven't seen anything like that.
Okay.
No, we don't know. And we really don't recommend or rely on the use of pesticides. Our graziers are not going to use insecticides on the areas that we're talking about. So sugar industry might, but again, we haven't seen any outbreaks in heavily treated sugarcane. So far, so good.
Okay, but yeah, sugarcane will be the place to look.
It will.
It's going to happen in Australia. Yeah. One more just came in. Does the mealybug like any volatiles in the plants like aldehyde?
We don't know. It's a good question, but no, we don't know. There's so much to do. I mean, I'm actually really happy that the Americans are involved in this now, because you guys will find out so much more than we could.
Well, unfortunately, we have a lot of need to get on it quickly. So
Absolutely. So, I'm handing it over. Here you go. Make the breakthroughs. We've got some work to do, but I think you guys have got the resources and the capacity to be able to really find out and build on this and get some hard answers to things that we can only say might.
So I know we've gotten to the end of the questions, which is crazy. That was awesome. I wanted to ask for all three of your perspectives, if I could, as we still got some people that have hung with us this long. As people are kind of going throughout their day and taking everything they've learned, can you give us just something that you would want people to remember about this, or just a piece of information from your perspective for people to take with them? So we'll start with Caroline.
Use a hand lens and look for the symptoms, and really learn to look for mealybugs. That's the most important part, is knowing that it's definitely the mealybug and that you know how to find it. And I will share the resources we have to help people to do that. And I think the other one is to remember it's a pest. We will get there.
Great. That's always something to remember, because especially when something pops up like this and everybody's trying to figure it out, we can handle it. We got it.
Yeah.
It's a great thing to remember.
Yeah.
What about you, Hannah? Do you have anything for people to know or remember?
Yeah, I guess I just agree. Like, so far it's not like a pest we've really seen before. And so we're just kind of keeping an open mind about how to move forward with it and building on, yeah, Caroline's work and other folks. And there's a lot of people doing work, and it's encouraging how many people are doing a lot really quickly based off of what they've been doing in Australia. So there is hope for as pain as much
Always.
as it's a pain right now. There's a lot going on.
Yep.
And Erin?
Yeah, I think it's awesome that we have so much like foundational work to build upon from Australia. And I guess just remembering that, like, we have different grasses and we're going to have to look at all of these different varieties and all of the different with different like climactic variables. But I think just not getting too bogged down on one path. Like, I think that Carrie's work has really shown like the importance of the endosymbionts and the soil health and all these things, and that it's going to require a diverse toolbox. And like, obviously the biological controls will take time, and looking into the physiology of the plant and the bug is also going to be really crucial. And potentially changing grass varieties and looking for susceptibility and resistance, but just keeping a diverse approach to this. Yeah.
And look, I think somebody said this has to be an international problem that's addressed internationally, and I fully agree. And if there's anything we can do to help you guys, or somebody wants to get hosted to come and see, let me know, and let me know soon as well, because I'm getting old. So it would be a real pleasure to help sort this out, because it's going to be a bigger problem even than the US and Australia.
Well, thank you so much to all three of you for coming together and making this webinar happen. I'm sure it's going to be so useful for everybody that's trying to just get their footing on what's going on with the Pasture Mealybug. So thank you all so very much. And
Thank you.
and I appreciate letting the Southern IPM Center have the opportunity to host. So thank you
Oh,
guys.
I'm really grateful to you for doing it and for inviting me. Thank you so much.